Hello, everyone. Thank you for being here today. We're very excited to have you join us, both from Australia and across the world. We've got quite some range of countries from around the world. And last time I looked at the registrations, we had over 360 of you today. So that's really thrilling because we're going to have a good time. So first of all, in the reconciliation, I'm going to get my slides to work. And I'd just like to acknowledge the traditional custodians throughout Australia from all the lands on which you were joining us from, and from the traditional custodians of all nations. We honour their connection to land, water, biodiversity and culture, and we pay our respects to their elders, past, present and emerging. So we're hosting this today. When I say we, I mean TERN, which is one of Australia's national research infrastructure projects. And our task is all about doing the monitoring of the changes to Australia's ecosystems so that we can advance this ecosystem science in Australia and contribute to sustainable management of Australia's environment. And TERN is actually not an organisation or institution. It's actually a collaboration of all of the universities of Australia and CSIRO, and we have also many international partners, collaborators these days. And together we carry out that incredible task of providing time series data from all sorts of different scales of measurement for people to use in decision-making, research, and just community understanding of the environment. And we are funded by the Australian government under an INCRIS grant. And today we are going to be talking about biodiversity modelling and does it matter? Does it work? And we have three incredible speakers. We're so lucky. They come from all parts of Australia and the world. The first speaker will be Dr. Martin Westgate, who leads ALA's Science and Decision Support team. And he specialises in tools for sharing and interpreting large data sets so that he helps all of us, I think, and he's a leading authority on quantifying biodiversity changes. I'm looking forward to hearing more about that one. Our second speaker is Jenna Wraith, who is the Senior Data Scientist at the Queensland Cyber Infrastructure Facility's Sustainable Futures team, and she's program managing a program that's called Wild Ops, which is about camera traps, and is a key contributor to the platform for modelling that's called EcoCommons. And amazingly, joining us at a very early hour in the morning from the UK is Louise McRae, very generously joining us. Louise is a research fellow at the Institute of Zoology at the Zoological Society of London and specialises in global biodiversity patterns and ecological monitoring. And she has been pivotal to the Living Planet Index. And some of you would have been at other webinars of TERN where we've told you how our Threatened Species Index is built upon all the excellent work that came out of making the Living Planet Index. There is a lot more about our three speakers in the bios that would have been circulated to you when you registered, so please have a look at those. I want to get straight into the talk, so I'm going to hand over to Martin to take us away. Thank you.
Lovely. Thank you, Beryl. Lovely to be here. I'll seek to share my screen. There we go. I'm going to assume that that's visible to everyone. Hello, everyone. Really glad to be here. Thanks for the invitation, Beryl and TERN. It's working for the Atlas of Living Australia, which is also an NCRIS facility, a sister facility to TERN. It's always nice to collaborate on these sorts of things, and I'm thrilled to be able to talk about the work we do. So, look, the brief for me was to sort of introduce the concept of biodiversity modelling from a data perspective. ALA is a data repository of sorts, a biodiversity database. And so I'll be talking about this from a data perspective and then handing over to Jenna to speak more in-depthly about modelling itself. Look, I'd just like to acknowledge that I've got the I'm very privileged to be able to live and work on Ngunnawal country. I'm in Sydney today, so a bit out of town, but we've got people from my team and from the Atlas spread all across Australia and several Indigenous nations there. So yeah, I'd also like to pay my respects to elders past and present in those lands. Now, look, I'm going to get straight into data aspects. I'm going to make some assumptions before I start talking about what we're specifically doing at ALA. So the first of those is that I'm going to be talking here about models that require data. Now, clearly that's not everything. You can build models that are purely mathematical. In biodiversity, that's not typically what we're doing. We're trying to study the environment. So I'm going to talk about models that require data. And I'm going to further make the assumption that that modeled observations should be alike. So things you wish to build a model from should in some ways be the same kinds of things. Now, this sounds like the sort of stuff that we all got taught in statistics class as undergrads, but I think it's an important principle here. Whether it remains true in future with the advent of AI is ambiguous and a bigger question, but I'm going to make the assumption that we want to model a single type of thing. And the last assumption I make in a lot of my work, though others would challenge it, I think code is a good thing when you're trying to work with the previous two assumptions. So we work at the ALA, we write codes in lots of languages. I particularly work in R. My team does development in R and Python. I think that helps with reproducibility and precision. And I think those are good principles. So that's my groundwork. And I won't talk about that again. What I am going to talk about is these four things. So I'm going to talk about what we do to standardize data, how you can get that data out of ALA, how you can, in future, we hope, publish data, and then what we're doing about the quality of data in the ALA. So look, just to get started, I'm going to use this phrase Darwin core. So this is not a concept I'd heard of before I joined the ALA. But it is nonetheless quite an old one and quite an important one to our organisation and to the Global Biodiversity Information Facility, for which we are the Australian node and for which many other countries also provide data. So Darwin Core is a data standard, a means of agreeing on what biodiversity information we should share. It's quite an old standard these days, and it makes this basic assumption that an observation of biodiversity requires place, a time, and a taxon. You can get away with without one or two of them, but really to make any useful inference, this is sort of a minimum. Some who know more about Darwin Core than I do may jump up and down and say, actually, this is a horribly minimalist interpretation of what that standard does. And it is. It's very, very brief. There's a lot more development on top of this concept. And how the Darwin Core standard works, but that's the basic principle. And so when I was talking before about my assumption being that we need an agreement about a set of things to investigate, this is the foundation of what we do. On top of that, then the way that people operationalize that in order to give data to the ALA or to GBIF or to other like facilities is to say, okay, well, those things can be organized in a few different ways. That basic type of data is what we would call an occurrence. Occurrences can occur within events. So let's say, for example, I went to a place, I started doing a survey, and then I might see many things, and those occurrences are nested within an event. I might take images, I might take video, I might take recordings. Those are all other forms of data. Those are all compatible with the Darwin Core standard. And then the only thing that that you need in order to package this up is two things that we wouldn't typically think about if you're just collecting data for yourself, but are very important if you're sharing it, which is something to say what that data is and how it's collected, a metadata statement, and what's called a schema, which basically just says, I've collected these three types of data and they join together in this way. Now, that's a like 40-second introduction to what Darwin Core is. If you have a profound interest in it, I suggest there's some very useful resources online. But we'll come back to this later on because this is important to the work that we do at ALA. So that's how we get data. How do we give it to you? So I've been at the ALA for 4 years now, and one of the first things I did there was to start building a piece of software called Galah. So Galar is an R and now Python interface to retrieving data from the ALA. And our principle here was to make that simple requirement that a lot of researchers have and a lot of decision makers have as easy as possible. And so what we've done is we've developed a syntax, which I've demonstrated here, for filtering the records that you want as quickly as possible, and then making very simple queries. So every function call, every call that you want to make to the ALA, you start with this simple function that just says, I want to start building a query. You build your query, which in this case is just a filter, but could have other facets to it as well. You can say, I want to count something, and then you just ask to retrieve it. So in this case, I've asked for a particular genus, and I've asked for those things that were seen by people rather than machines, and rather than specimens in museums. And I find that's the number of them that the ALA has records for. I can then amend this query. In this case, I've added an extra filter to say I just want them for the most recent year. And I've removed the count request here so that rather than getting just a summary of the number of records, I've just asked for those records to be returned. And assuming I've signed up for the ALA and have a login there, I can then retrieve that data seamlessly into my workspace and get some information about and this is what Darwin Core looks like when it's operationalized. These are our basic fields, and it's fairly obvious: a decimal latitude, a decimal longitude, scientific name. Some of it's a little bit more technical and in some ways uninteresting. So things like we give each taxon an identifier, or rather we use identifiers from a national species list. That is important for us programmatically, but it's not especially, it doesn't make any difference. It's just a code that represents the scientific name that we're provided. And so we feel that in providing as clear and as easy to use a syntax as possible, combined with as clean documentation as we can provide or think of, that we're lowering the barrier to entry to all these kinds of data. Just to give an extra example of that, you don't just have to count the number of records. You can say, actually, well, what I want to do is find out how many species I've got. So it turns out for that same query, we have records from 7 species. And you can then say, well, actually, I don't want to count them. I want you to show me what those species are. And you can get a list of that too. So we've put a lot of thought in this. We're pretty proud of it. I hope it's, as far as it goes, we think it does a good job. We've deliberately used a modern syntax called tidyverse, which is to lower the requirement for people to learn new syntax to use our software or use our database. We've developed as clear documentation as we can to try and make that more accessible. And the last thing we've done is, because the Atlas of Living Australia is just one representative of several organizations that all use this Darwin Core data standard, we've tried to make the software call all those organizations as well. So for example, you can call all of these different locations just by changing a single parameter within Galar. Just say, point this at GBIF, point it at the UK, point it at France. And then all your subsequent queries give you information for a different database in the same standard. And that is, of course, the benefit of a standard, is that if everything's formatted in the same way, then the queries you send are interpretable by a range of different organizations, and the data that you retrieve is similarly comparable. So our latest activity in trying to support users to do useful things with the Darwin Core standard and with the ALA and with GBIF is called Galaxius, and this has not been released. So the code I'm showing you here is experimental. You can download it and use it right now if you want off our GitHub page. It'll almost certainly break, but we're hoping within the next weeks and months that will be less the case and ready for launch. So what we're trying to do here is recognize that actually several different types of groups collect biodiversity data of the type that we've talked about today. And rather than simply downloading it from us, maybe there's a desire to contribute to that database as well. Now, we're a voluntary organization. We don't own any of the data we store. If people want to send us data, we'll publish it, but it remains the property of the people who provide it. But nonetheless, there are some barriers to providing data to us, and they're technical. I mentioned before that the Darwin Core Standard wasn't something I'd heard about before I joined the ALA, and I think that's, if not universal, fairly common. And so we've built some software that we hope lowers those barriers to entry, just such that if people do want to share data, that's easy to do. And we have broken down into the following steps. So we encourage people, if they want to do that, to set up a new code project. They need to match their data to the Darwin Core terms or the headers that we use as a standard. They need to add their own metadata, and then they need to validate and publish that data with us or with another organization. Obviously, publishing is always a voluntary stage here. So just to give an example of what we've built and how we hope that'll be useful to people, this is just an example of what the sort of information might look like if it was sitting on your desktop or on your machine somewhere. We have some quite simple functions. In this case, it's currently called suggest workflow. It's in flux at the moment. That simply says, look, haven't matched any of those names to anything that in the Darwin Core Standard, but these are some things that you might want to consider sending us. I found out just before this that the output that you see in your console doesn't render properly in this sort of slide. So actually what is shown when you do this in real time, you actually get a suggested workflow that says, hey, if you use these functions, then you'll end up with a Darwin Core compliant data set. For some reason, part of that is wiped in this slide. So my apologies there. Clearly some kinks to iron out there. But the point being then that people don't have to think or even really know about the standard to be able to convert data into that standard in a quick and easy manner. That a lot of those steps are provided for them and bespoke to the information they've provided. The other challenge we've had is that metadata in the Darwin Core Standard has to be provided in a format called XML, which is HTML-like, but it's hard to write from scratch. And so what we've done is provided a new package, which is called BioGalaxis, called Elm. Elm is nearly ready to go, which takes either XML and converts it to Markdown or Markdown to XML. Now, that might sound no less technical, but just to give an example of what I put here, we store metadata that people give us for their own data on our own system. So this is the URL to a metadata standard, in this case from the New South Wales Bionet Atlas. You can download that directly using this software, and it converts it into a tibble in R. And then you can export that in a single step into this format. Now, this doesn't look very pretty, but the rest of it is human readable. You write that out as a Markdown file, and that can be rendered to a PDF report, or critically, it can be altered and put back into XML using the same software. So you could literally take this as your example and say, well, my name isn't New South Wales Bionet Atlas, it's some other organization. So use this as a template, for example, convert it back into XML and submit your own data. And you could use that using any existing XML that you can find, or any of the 300 data providers that have provided them to the ALA. So what we're hoping then is, while this is still looks, perhaps not as pretty as a Word document, it's human and machine readable and interchangeable. And the benefit of having that in a coding environment then is that it is an entirely reproducible solution. So just to prove that happens, you can take that metadata statement, convert it back into an XML and do that in simply, this line here is just for display purposes, but this one function converts it into the standard that we need to submit data to the ALA, GBIF, or elsewhere. So that's the basics. Having a standard, getting data out of that standard, putting data into that standard. And we're very close to sort of closing that loop. And what we hope then is that the combination of tools will help grow the community around sharing data using that standard, and therefore understanding more about the environment. There is a last step to this, which is about the quality of data. And that's a term that gets used a lot. I think it's a challenging one for us at ALA because we're storing 150 million observations from, you know, 300 or 400 years worth of history, obviously there's points of difference among data collections, among years, among taxa, as to what that data can be used for. And our current system is to have a group of what are called assertions. So we check records. So this is simply a call, again, demonstrating our own tool set here. You can find out how many records of the 150 odd million that are in the ALA have particular assertions. Now you'll see that actually there's most records are, if not perfect, pretty much fine according to our technical checks, but a small number of checks give quite high numbers of records. And just to give a demonstration of roughly what that means, in spatial terms, we are often missing georeference information, such as the coordinate reference system that data were collected in. In taxonomic terms, we're often missing, we've got taxonomic names, but not their rank. In terms of observations, we're having to infer one field from another. Our temporal data is actually extremely clean. One of the biggest flags we get is that something's on the first of the month, which is it can be a flag that someone didn't know what day something was collected, but you would also expect to see in about 1/30 of all records, even if they did know what date it was collected, and that's about what we find. So this isn't, so several of these are either are very small problems around people having information but not sharing it in quite the way the standard expects. And so we're trying to help work with software, our data providers, and our users to sort of clarify those points of contention, which isn't to say that would solve all of our problems, but it's showing that there is a base level of quality there to build on. In terms of next steps, we're auditing that system. So we're interested in working out how it can perform better, and particularly how it can start to do more mathematical or statistical approaches to infer high or low quality data according to different facets of what that might mean to different people. So a good example here, and I've mentioned error type classification. What I'm talking about there is we find, for example, that older records tend to have a very good taxonomic description, but sometimes are lacking in their spatial description because they were simply recorded before a time where that was easy to find. Whereas modern records may have more, and some facets of the citizen science community have this problem, not all of them, where taxonomic certainty can be a bit lower in some cases, but the spatial precision is high because people are recording things on their smartphones. So building some sort of statistical approach that can account for that sort of diversity of provider and look for problems like outliers given that diversity is a challenging statistical problem, but one that is going to be important for us to help our users and our data providers in future. The other thing I'm going to raise briefly is this thing called event data. So I mentioned before that the Darwin Core schema enables people to group observations together on the basis that they were recorded at the same time. That's critical for things like presence absence, to store presence absence data properly, which is similarly important for occupancy modeling, time series analysis, building things like threatened species index. Or many extensions of ecological modeling depend on that sort of data structure. And we're working with our global partners, GBIF, to try and build a model that can support that within the ALA, which I think would be a real game changer for our utility for ecological modeling and for ecological inference. I'll put a plug in as well for this resource that we've just developed, which is a tool for those using ALA, data from ALA or similar resources who find a need to clean data themselves. It's entirely in R, it's online. Google this title, you should find it. It's written by my team, but led by Dax Kelly, my colleague at ALA. Check it out, there's a lot of useful tips and tricks in there. I'll stop there and say thanks to my team and encourage you to check out our website at labs.ala.org.au. And with that, I will hand over to Jenna to talk in more detail about the modelling side of this question. Thank you.
Awesome. Thank you, Martin. That was a great talk. Okay, I'm just going to share my screen. Okay. All right, I think that looks good. Hi, everyone. Thank you so much for having me here today. I was so excited when Beryl invited me to talk at this webinar. It's always such a great event and awesome to see people from all over the world. So I'm Dr. Jenna Wraith, and I'm a senior data scientist at the Queensland Cyber Infrastructure Foundation. And I work largely on the EcoCommons platform and also manage the Wild Ops project. And today I'm going to be talking to you about the role of biodiversity modelling in environmental research and management and how the EcoCommons platform is helping to drive that forward. But before I start, I would first like to acknowledge the traditional custodians on the land in which I'm meeting you from today, which is Yugambeh country on the Gold Coast. And I pay respect to elders past, present and emerging and extend that respect to all Aboriginal and Torres Strait Islander people. And of course, I couldn't get started talking about the EcoCommons platform without first acknowledging that the EcoCommons platform has been a huge collaboration with some awesome partners. So we're a joint initiative and we are partnered with ARDC and these other awesome partners here. And as I mentioned, I'm working at QSIF, and the EcoCommons platform sits in the Sustainable Futures portfolio. And in this group, we support projects that range from environment, biodiversity, agriculture, and urban design. So the EcoCommons platform has found a really great home here. So now I'm going to get into biodiversity modelling, which is essentially all about predicting how species, distributions, populations, and ecosystems might change over time or under different environmental conditions. These models are built using a variety of data inputs, such as species occurrence records, like those that Martin just talked about from the ALA, or environmental variables like temperature and rainfall, and even human activities. And these applications that come from these models are really vast. They can range from conservation planning, habitat restoration, to environmental impact assessment, and supporting policy and decision making. So why is biodiversity modelling so important? These models help to provide critical insights that inform decision making at all different levels. This can be from local conservation efforts all the way to some big international policies. And by helping us to understand and predict the effects of environmental changes like climate change, habitat loss, and pollution, biodiversity models really enable a more strategic conservation planning, and they help to ensure that our resources, which I think we all know are often quite limited, are used in the most effective and efficient way to protect our natural world. So as we all know, we are facing a multitude of environmental challenges at the moment that make biodiversity modelling even more important. We have climate change, shifting species distributions, and altering ecosystem dynamics, while habitat loss and fragmentation are leading to reduced biodiversity. We have invasive species that are often outcompeting our native species, which drastically changes the ecosystems. And we also have things like pollution having a huge impact on species health and ecosystem resilience. So biodiversity models can be really powerful tools to help address some of these challenges. Their predictive capabilities allow us to forecast how species and ecosystems might respond to various environmental challenges. The information is really valuable for targeting those conservation efforts and actions, and ensure that we focus these efforts on areas and species that need it most. Also, these models provide the data-driven evidence that's often extremely necessary to support environmental regulations and policymaking. But as we know, biodiversity modelling is certainly not without its challenges. So one of the main issues is the quality and availability of data, which can often have gaps and inconsistencies, which can result in unreliable models. We are lucky that we have data sources like the ALA that Martin talked about that really helped push this forward. But sometimes when we do have data, it's often difficult to integrate diverse data sets together. So it can be difficult to incorporate spatial and temporal data into cohesive models, which can become quite a significant technical challenge. We have very complex ecosystems that we're working with. They have intricate interactions and processes, which adds a layer of difficulty when doing these models. And now we also have the need for increased high compute power to actually run our models. So this can be due to increasing fine resolution data which is awesome. It's so good we have this new data source, like satellite imagery, we have drone imagery, and we have wildlife camera imagery, all of which can be fine scale, but requires a lot of compute power to actually use in our models. And a lot of these models require advanced coding skills. Like Martin showed, there is a need for skills in R to do some of these things, and also increasingly in Python. So at the EcoCommons team, we conducted a survey to look at what our users, so students, researchers, policymakers, and industry workers in the ecological space, what their needs and barriers are when running ecological models. So we can see that in this space, there is a need for SDMs and climate change experiments. There was an emphasis on the types of models, which were generalized linear models and bioclim algorithms. There was a real need to be able to share data in an ethical and secure way, and also a need to have more guidance so people can choose the best data and models that are fit for purpose for their needs. Also, compute power was a big one, and more collaboration, which I think we all appreciate. And the barriers, they did match these needs as well. So, a lack of training and support, the level of coding required to run a lot of these models, the compute power, and the data wrangling, which is such a big technical challenge. So we took these needs and these barriers, and we used that to guide the development of the EcoCommons platform. So we started by including access to thousands of curated data sets, including access directly from our partners at the ALA and also at TERN, among others. And then we built the platform that allows users to run these models on cloud computing. And so minimizing, you don't have to run that on your local computer, it all runs in the cloud, removing these technical barriers without the need to code as well. And we also want to create a community of practice to improve biodiversity modelling in research and decision making. So this brings me to the EcoCommons platform today. It's a platform built with the specific needs and challenges of our community. Beyond addressing individual needs, the EcoCommons is designed to be a collaborative space where researchers, policymakers, and practitioners can work together and build and define biodiversity models. The platform has seamless access to these integrated data sets, advanced modelling tools, and scalable workflows to cater for both beginners and advanced modelers alike. And our goal is not just to provide the tools, but also foster a community that advances ecological research and decision making. So some of the key features of our platform include a range of species distribution modelling algorithms. We have around 20, and these allow users to predict species distributions under various environmental scenarios and also predict these to future climate scenarios. We have our Data Explorer, which is a really powerful tool for assessing and analysing a wide range of data sets, and you can visualise them too. And we're building a library of Jupyter notebooks to enable users to create and share custom workflows or use ones that we're developing as well. We also have access to peer-reviewed education materials and platform guides so that all users have the tools that they need to start modelling the best way. And the platform supports collaborative workspaces, making it easier to collaborate with your colleagues, share workflows, and discuss results. Now I'm just going to play a quick demo. Yeah. We're going to play. Okay, so just to show you what the platform looks like, this is where you can see a range of 60,000 data sets. You can search using different filters. You can view these data sets here and see the metadata. You can import data from third parties like ALA, GBIF, and TERN. You can also import your own data if you like. And you can run your models here in the BCCVL, where we have a range of different species models and climate change projection experiments. And running an SDM is as simple as clicking through a series of tabs here, adding your data that you've imported. You can visualize that here. You can select the way that you want to use your absence data or pseudo-absence data, your climate and environmental variables, and you can use different ways to select your study area, including a range of predefined areas. And then we have our series of around 20 algorithms that you can choose from, running multiple at a time. You can edit all the advanced settings that you would if you were using the R packages, and it runs in the cloud. And then all the results are available to you to visualize on the platform, or you can download them as well. And if we visualize them here, you can see we have maps, plots, CSVs. We provide all the metadata and the code as well. So everything is repeatable and transparent. And then we also have a function that I really love, which is to be able to share your workflow with colleagues. So you can put their email in and share this experiment. Okay. So what's next for the EcoCommons? So we're really excited. Now we're sitting at QCIF. We're looking at incorporating a range of different data sets. We have new data coming from satellite imagery, drone data, and wildlife cameras. And we also want to focus on incorporating more marine data. We have a lot of terrestrial. So if you have some marine data, please get in touch. We also are working on expanding our services and adding new functionalities, new models, and we're also building our Jupyter Notebook library, where you can create and share your own modeling workflows or use the ones from us. And we're also working on integrations with other platforms. For example, the Wildlife Observatory of Australia, which also sits at QCIF, which will provide access to wildlife camera images that have used computer vision to automatically detect which species are in those images. And that will flow on to the Atlas of Living Australia as well as to EcoCommons, which is really exciting. So how can you get in touch with us at EcoCommons and get started? So you can register on the EcoCommons platform at ecocommons.org.au. You can register using AAF if you're at an Australian institution, or we can create an account for you. You can attend one of our upcoming workshops. So we have workshops at the Ecological Society of Australia conference this year and also the Queensland ResBaz. And please share EcoCommons with your network. And just to finish up, the EcoCommons is providing access to thousands of users across different sectors. These range from government, industry, and research. And the service and its previous iterations, which included the EcoCloud and BCCBL, have been used to answer thousands of questions and research projects from marine to terrestrial applications. Our two key features, our datasets and our models, offer enough flexibility and data intelligence to answer the environmental questions that our users have. So the EcoCommons has become more than just a platform. It's a catalyst for innovation in research and policy. By providing high quality tools and data, the EcoCommons helps inform evidence-based decisions that are really critical for conservation and environmental management. And that's all from me. Thank you so much. I'm now going to pass on to Louise.
Great. Thanks very much, Jenna. That was a really fascinating talk and great to hear about EcoCommons. That looks like a brilliant tool. Hi, everyone. It's really nice to be here. I'm just going to bring up my presentation. So, yeah, my name's Louise McRae. I'm based at the Institute of Zoology at the Zoological Society of London. So I'm joining you from the UK today. And I'm going to focus my talk more on some of the outputs of biodiversity modelling. So I'm going to look through the lens of biodiversity indicators. And specifically, I'm going to talk about one I've been focused a lot on, which is the Living Planet Index. So I explain where the data come from, how we model it, some of the applications that we use, some of the limitations that come from the data and methods, and then a couple of examples looking ahead at how modelling can help us overcome some of those limitations and improve our understanding of biodiversity change. So just a brief summary of biodiversity indicators. These summarize complex data often and trying to convey it in a very simple, communicable figure. So because biodiversity itself is complex, no single measure can adequately describe the whole of biodiversity. So if we just take species as one example, we could look at how abundant a species species is, what the risk of extinction is, how it changes in relation to the community it's in the composition, and how its distribution might change over time. So there are many different measures just for species. And we can also look, this figure on the right-hand side is quite old now, but shows you the different types of biodiversity indicators that can be used. So they can also be used to look at changes in the state of biodiversity, pressures, the response and benefits that we get from biodiversity. So the state might be something like the red list index or the extent of forest. Pressures could be the increase in the number of alien species or proportion of fish stocks overexploited. Responses are something like the increase in the extent of protected areas. And the benefits are trends in species that are useful for food and medicine, for example. So the one that I'm focusing on is the Living Planet Index, and that's an indicator of the state of biodiversity globally. This index shows the average change in relative abundance from almost 5,500 vertebrate species and over 40,000 populations. What I mean by relative abundance is that we look at the rate of change in each population rather than the absolute change in the population size. Now, the Living Planet Index, or LPI, which I'll refer to it to just save time, is published biannually in WWF's Living Planet report, and it's been coming out in that report for every 2 years over the past 25 years. Now, why population data? Well, population data can be quite powerful. We've got very fine scale data which measures population over time. Now, this can give us an early warning of extinction risk. It's because we've got a specific location and it's measured over time, that gives us that sensitivity that we can start to understand how this population is impacted by different drivers of change. And we can also see what happens, how populations respond to conservation actions as well. And broadly, populations can often indicate the health and resilience of ecosystems that they occur in. This map shows the locations of all the monitoring sites that are used in the Living Planet Index. And at each of the sites, different monitoring approaches are taken. So this will depend on what type of species it is that's being monitored. So it could be using nets if it's fish, there could be aerial surveys, a traditional walking transect using visual sightings, or perhaps often using sensor technologies like camera traps, for example, for some more elusive species. And I'll use the term population a lot. That's a bit of a shortcut for saying a species monitored at a particular location over time. I'm not meaning population always in an ecological sense. So how do we get from the data to the index? Well, the data are collected in the field, they're often then processed and published, and then the team based at ZSL search for the data and then add this to the database if it passes the set of criteria. So we follow a fairly simple protocol for adding data, and as long as a standardized method was used over the time period, then we can incorporate it into the database. And as you can see from here, we can incorporate many different types of data as long as it represents the abundance or population size of that population. So, for example, we can use densities, biomass, different indices, or perhaps some sort of proxy. For example, for birds, we often use pairs or nests. And because that range of different measures that we are incorporating is is so broad, that gives us some unique challenges to how we then process the data. So firstly, we've got different units, as I mentioned, of measure, and we also have different scales. So some of the locations are quite small. It might be a patch of forest, and then other sites could be much larger, entire national parks, for example. And the populations themselves are of different sizes. We have island endemics, and then we have very common species numbered in the many thousands. And also because monitoring is not standardized across the whole planet, we have variation in the duration and the frequency of these time series. So they don't all start and end in the same year. And also, as you'll see from this figure on the right-hand side, the taxonomic and geographic representation is quite patchy in places, and we tend to find that a lot of the data comes from more well-monitored, traditionally more well-monitored regions, and we do lack gaps in areas where we have high species richness. So the darker background colours on this figure, the browns and the darker blues, show the regions of land and the oceans that are higher in species richness, and you can see that there are some gaps in the data set. So this is how we go through the process of modeling the data. So the first step is using a generalized additive model framework. And we have two different options for how we model the data. And this is based on how many data points we have in each population time series. So we need to, this gives us flexibility. So it means that we can incorporate populations that only have 5 or fewer time points within a time series. But a time series needs to have 6 or more data points in order for us to be able to use a generalized additive model. And using this model enables us to capture long-term trends. So it will reduce the sort of interannual noise that you can often get from monitoring. So that's the first stage in processing the data. And then next, when we've modeled the population trends, we then aggregate the index from the population level to give a species trend. And so, for example, this bird species on the left-hand side, we've got 4 different populations at 4 different locations, and these trends are averaged to give the overall bird trend. For this primate, we only have a single example of a population, and so that will provide the species trend for that species. And then all of the species are averaged together, and that produces the overall index. And that's the simple basic building blocks of the Living Planet Index. When it comes to the global version of the index, we introduced this third step, which involves proportional weighting. The reason for this is going back to what I said before. This is just a different version of that figure I showed, where we've got species richness as the base layer in the land and on the sea, and then those locations show the locations where we've got LPI data. And because of this imbalance, we try and proportionally weight the global index to capture the areas of higher species richness. So, for example, the highest weight in the terrestrial and freshwater Living Planet Index is given to the neotropics, as this is the region that has the largest number of species that are known to occur there. The lowest amount of weight is in the Nearctic realm, as that has the lowest number of these species. And in the marine species, it's the tropical Indo-Pacific that has the highest weighting. And then the Arctic has the lowest weighting. This is not the perfect solution to this, and I'll touch on this a little bit more, but this helps us turn this sort of noisy and uneven data into a simple figure that's designed to indicate a global trend. Now, some of the outputs of the Living Planet Index, as Beryl has mentioned, and has been mentioned in another talk, is the Threatened Species Index. That's how that uses the framework of the Living Planet Index, and that's used at a national level for policy. And the LPI is also used globally as well. So the Intergovernmental Science Policy Platform on Biodiversity and Ecosystem Services. That's quite a mouthful. And the LPI has been used in a number of those global assessments. Oh, beg your pardon, I'm just going to go back. And then also it's used for communication. So it can convey quite a complex topic of biodiversity change into a single message for a broad audience. As I mentioned at the start, the Living Planet report is the main avenue for the new updates of the LPI, and it's translated into many languages and circulated around the world. So it does have quite a broad reach for a broad audience. However, there are limitations. Using a single number masks complexity of biodiversity change. So we know that not all species are in decline, and we know there's a lot of variation. So this figure on the right shows that trends vary according to the different regions around the world, according to different habitats and different taxonomic groups. And there's also some limitations to the proportional weighting approach, as I mentioned. So this figure shows on the x-axis the estimated number of species known to occur and the number of species included in the LPI. So the perfect representation would be up here, would be this 99%, and the lowest representation is down here, the 1%. And if I just show you, the green and the purple show the Nearctic and the Palearctic representation, which tends to be better. We tend to find that mammals and birds are much better represented. Generally, the poorer representation comes in the more tropical regions, shown in the blue and the slime and the orange, and also in fish and reptiles and amphibians. So I'll just end on trying to mention a few approaches where we're trying to use modelling to deal with these limitations. So firstly, we've got explanatory models. So these can use land use change data, climate warming data, to then explain why we're seeing different population trends. So that's one approach. And because we've got this spatially explicit information, we're able to combine those data. That then helps us then develop predictive models. So this will allow us to fill in the different pieces of the puzzle and try and make predictions of what's happening in areas that we don't yet have data for. And then finally, this can also help us to look ahead. So this paper came out a few years ago, combined lots of different biodiversity models and indicators, showing that historically, biodiversity has declined, and with business as usual in grey, is predicted to continue to decline. With increased conservation efforts, that trend is predicted to stabilise and start to increase. But if there were increased conservation efforts and the way we produce and consume food was made more sustainable, that would start to actually show some positive trends. So I hope I've given you a bit of a flavour of what the Living Planet Index is used for, how we use time series modelling within that, and how we might be able to use different explanatory models and predictive models to overcome some of the limitations. So thanks very much again for Beryl for the kind invitation. I'd like to acknowledge my funders and collaborators and the rest of my team at ZSL, and please reach out if you have any questions. So I'll pass now back to Beryl. Thank you.
Thanks, Louise. Fantastic talks from you and Martin and Jenna. It's a highly technical subject, but I managed to keep pace, which is really good. So well done, everyone, for superlative communication. There is a question sitting in Q&A, Louise, which you haven't had time to look at, which is, does the Living Planet Index consider shifting baseline syndrome? You'll have to tell me what that is before you answer it.
Yeah, that's it. So yeah, shifting baseline syndrome is essentially where, where your what your expectations of what nature or biodiversity should be shifts, I guess, over time. So what I might think of as intact nature might be different from my parents and my grandparents, for example. And so it can be a problem because we set the Living Planet Index to start from 1970, and that's in a sense an arbitrary baseline. The reason is because that's when we find we've got most monitoring data that it starts from. There's very little data prior to 1970, but that does mean that we're only comparing to 1970. And if you think about, for example, in Europe, where I'm based, a lot of the transformation to land and to species happened prior to that. So we're not necessarily seeing the full picture of what's happened sort of since humans have started impacting the environment.
Thanks. And Eddie Cannell has actually posed one for all three of you to have a go at, which is how often are the models revisited to assess their relevance and efficacy, and what can be done to the seemingly increasing use of models as absolute predictors rather than suggestions of what may be the case. Who wants to start on that one?
I guess I can add something to that. In terms of the EcoCommons platform, for their relevance and efficiency, we've updated our models quite regularly. We have scientific advice. We're built on a scientific advisory committee who have put a lot of time and effort to update those models, algorithms, and the code that they're built on to make sure they're up to date. And that's something that we iteratively work on. So it's, we're always hoping to be using the best practice. In terms of being absolute predictors, I think models always need to be a supportive part of the process. I think there always needs to be, collaboration with land managers, traditional custodians of the land, and, expert knowledge on the systems. I don't think they can be the whole story. I think they're just part of the story.
Any other?
Anyone else?
That was good. Thanks, Jenna, for that. Any other comments?
I'd just back that up a bit, actually, just in the sense that, look, it's funny, we talk a lot about modelling at ALA. We don't do a lot necessarily, but we have real problems because for something like 50% of the taxa that we know are in Australia, we don't have a single record in the atlas. And that means that almost no museum or collection in the country has one or has digitised it yet. And to a greater or lesser extent, that's a problem for some taxa everywhere. And I think that was really well shown by Louise's talk, that for some taxa, for some places, we're doing amazing work and we're getting better all the time. But for some things, we don't know. And so actually, that's a problem that modelling can help with to some extent, but only to some extent. And for some things, we don't know. It's just not the information.
It's good. And everyone that can access the Q&A, you'll see that Louise has popped in the link to the Nature paper that was well done for that. Louise, can you just confirm that there was a question that does the Australian TSX use exactly the same methods for tracking trends?
Yeah, good question. Yes, the framework is pretty much the same. I would say the difference is the threatened species index focuses on threatened species only, nationally threatened species in Australia, whereas the Living Planet Index encompasses all and any species. But the basis of how the data are modelled and aggregated to the index is the same. The TSX doesn't use any proportional weighting. So what I explained for the global, that isn't incorporated. But yes, and excitingly, the TSX includes plants, which the LPI does not. So that's very exciting.
We do have other questions, things about using Galar for overseas data, and also there were lots of questions about putting up your own data in EcoCommons, but I think we'll have to tackle those outside of this meeting as we get to the end. It's been a fabulous series of chats going from the data to the models to the decision making that's possible from it, everything that we actually wanted out of this topic. And I'm just going to share a screen and say we've actually got more exciting things coming up before the end of the year. We have one more webinar for the end of the year, and it's all about drought, particularly ecosystem drought, and all different facets about that one, because TERN is involved in an international program on that. And now I just want to wish everyone to enjoy the rest of their day. And thank you all. And if you have any questions or feedback, please contact us again. And we wish you goodbye. Thank you.